Thank you. Well, good afternoon. It's a pleasure to be here. And the idea behind these 2 hours that I'm going to be speaking is basically to try to convince you that if fMRI was what it was supposed to be when it was born 20 years ago, it certainly wouldn't be able to survive as it did. So what I want to convince you is that fMRI has changed. fMRI is not the technique that's capable of mapping different structures in the brain that respond to specific stimuli. It does it too, but it's not only that anymore. So my perspective here is to walk you through part of this history, from where it begins to where is it right now. The first idea behind fMRI was to try to overcome this only access that we had to human brain that was by means of invasive measurements. And that is not, I mean, we were capable of doing this kind of measurements only in the 30s. 1930, we're capable of studying the electrical activity of the human brain in, of course, in a limited number of subjects, because we needed, the only brains that we were capable to inspect using these techniques were patients who were going to surgery. Although this was the case, there are many different non-invasive techniques that are becoming more and more available to research. And here I just have a couple of examples. The most traditional and the most known one is EEG. All these techniques, they have advantages and disadvantages over one another. Some of them have a very high temporal resolution. Some of them have a very low temporal resolution, and on the other hand, it has a good spatial resolution. Some of them are capable of detecting the electrical activity itself. Some of them are not capable of detecting the electrical activity. They have to rely on an epiphenomenon, like cerebral blood flow or something like that, or glucose metabolism, or any epiphenomenon that's behind the neuronal activity. The EEG, on one hand, is one of these techniques that's capable of detecting the electric activity from the neurons, but it has a very limited temporal resolution. And the reason being is that, spatial resolution, I'm sorry. And the reason being is that it is very sensitive to different conductivity of the tissues once it leaves the cortex surface all the way up to where the electrode is. It has to go through different layers, and every layer has a different conductivity. And EEG is very sensitive to the conductivity issues, meaning that you know when that event happened, but you don't know where this event is coming from. On the other side of the spectrum, we have fMRI. And that is the technique that I'm going to be talking a little bit longer today. And the main problem behind fMRI is the fact that we are not detecting the electric activity of the neurons, but we are detecting the increase in cerebral blood flow as a response to the increase of neuronal activity. I'm just going to walk through some of these techniques, as I'm not going to talk about them, but I believe it would be interesting to walk a little bit on these different techniques. On the middle here, we have MEG, magnetoencephalography. And the idea behind MEG is exactly, it's basically the same idea that's behind EEG, but what you're detecting is the magnetic field that's produced by the varying electric field produced by the neurons. So basically what you're doing, you're detecting the electrical activity of the neurons. The main advantage of the MEG over the EEG is the fact that magnetic field is not sensitive to conductivity issues. Meaning that you have, with MEG, you have a much greater spatial resolution as far as detecting where the sources of that activity is at. What is the main disadvantage on the MEG is the price, basically. MEG works with a special kind of device called SQUID, that stands for Superconductor Quantum Interference Devices. As the name says, these detectors, these devices, they are superconductor devices. So in order for them to operate, you need to have them inside a liquid helium vessel. And that's where, this is what this is. This is a liquid helium vessel. The detectors are right here. So you cool down the detectors, you cool down these squids, and then you're capable of having this system, these squids detecting that activity, that magnetic field. And the other problem is that these magnetic fields, they are very, as they are very sensitive, they need to be inside. This whole system needs to be inside of a magnetic shielded room that are expensive. To buy a system like this one, you would spend something around $2 million to $2.5 million, the whole system. And the operational cost of this system is high as well, because some of this system, they consume about 20 to 30 liters of helium a week. And we are talking about a liter of helium, you're talking about $20, around $20. So you do the math. It's not a cheap system. And what you increase when you compare an MEG with EEG is the spatial resolution that the MEG has, which is much greater than the EEG. Down here, we have this whole new line, line of research and line of clinical application. That's the nuclear medicine techniques. Basically, we're talking about SPECT, that's single photon emission computed tomography, and PET, positron emission tomography. The spatial resolution and the temporal resolution of the nuclear medicine techniques are very low, both of them. Then what is the reason that, so spatial resolution and temporal resolution are not the only issues behind the techniques that you're going to choose in order to have your question answered. The greatest advantage of the SPECT and PET is the fact that you're capable of tagging a specific substance that you're interested in. So you're capable of tagging glucose if you're interested in mapping glucose metabolism. You're capable of tagging serotonin receptors. You're capable of tagging dopamine receptors. So you're capable of tagging different substances, and then you're capable of pretty much localizing where those substances are concentrated in non-invasively in the human brain. So this opens a perspective, like a neurochemistry perspective, on looking at the brain and trying to understand the chemistry and the pharmacological issues behind the human brain. These techniques, or near-infrared spectroscopy, they have been released a couple of, maybe 10 years ago in a clinical setting. And the idea behind this technique here is very simple. It's similar to the one that we use when we measure on oximetry. So basically what you do is you emit infrared light and you detect the infrared light that got scattered by the tissue. Since hemoglobin, the total hemoglobin, oxyhemoglobin, and deoxyhemoglobin, they have different spectra, absorption spectra, then you're capable of measuring the concentration of oxyhemoglobin, deoxyhemoglobin, and total hemoglobin. The spatial resolution is very limited, is very superficial, and where is it that it found most of its application in children? So a lot of neuroscience has been done in children using this technique, using NIRS. It's very simple to put on. It's really straightforward. If you compare that to an EEG, that seems something that's similar. Placing a 64 EEG system takes about 40 minutes. Placing an ear infrared system takes about 2 minutes. It moves with the head. It's not sensitive to motion artifact as the EEG system is. So it's a really interesting technique if you're interested in working with kids. This last technique, I believe that Claudia is going to talk about when she comes on Wednesday or Thursday, I'm not sure. That's for the transcranial magnetic stimulation. So what you're doing with this technique is looking the other way around. You're not detecting the activity itself. You're stimulating specific parts of the brain and looking for a response. There are two main applications behind the TMS. One of them is what we call a single pulse. And with a single pulse, you just have a single magnetic pulse. So you have a magnetic pulse that's going into the brain, and then it stimulates specific areas of the brain, and then you look at the response. Most of the TMS studies for single pulse are done with motor system, or done to inspect the motor system. And then you have the repetitive TMS, or RTMS, that you have a train of pulse of around 1 hertz, and then with doing that, you're capable either of hyper-exciting specific parts of the brain or down-modulating specific parts of your brain. And then this has opened up a perspective of doing what we call virtual lesions. So you lesion, between quotes, specific parts of the brain, and then you measure performance. And then you have an indirect way of measuring how important is that specific part of the brain to have when you perform a specific task. And then once you turn off the train of pulse, then you're done. I mean, the lesion is done. So it's reversible, it's safe. So it's a nice strategy if you want to move the other way around. With this slide, the message I want to bring is the fact that all these techniques, they have advantages and disadvantages. So a lot of the studies that we conduct, they rely on the need to use more than one technique. So a lot of studies, you can use fMRI together with TMS, you can use TMS together with EEG, EEG with fMRI. So you can have a whole bunch of combinations between these measurements in order to have a more complementary way of looking at your problem. So talking about fMRI, so the whole rest of these 1.5 hours, I'll be speaking about fMRI. So what is fMRI? For those of you who don't know fMRI, I decided to go through these very basic steps where it came from. And this is what we need to know as far as what happens metabolically, what happens as far as energy consumption in the brain. If we split the process of many process of neuronal communication, and we try to look at how much energy does that process consume. So you see that most of the energy that's consumed is used to restore the potential after an actual potential. So restoring an actual potential is something that consumes a lot of energy. And another great part of the energy is consumed as a restoration of IPSP and EPSP, inhibitory and excitatory postsynaptic potentials. So we see that about 80 to 90% of the energy consumed, 85% of the energy consumed in the brain is consumed in processes that are important in neuronal communication. So where does this energy comes from? The human brain doesn't produce glucose, it doesn't produce oxygen. So the way we bring that energy into our brain is through our vascular system. So the vascular system, the information is important right now, is to know that the vascular system, it is organized in different scales. The first scale, or the vessels that are of great caliber, like the arteries, they are not capable, or they have a limited capability of, they don't have muscles, so they're not really keen at doing vasoregulation. So they do not dilate, they do not constrict. So they are pretty much vessels that are capable of taking the blood all the way up to the brain. Once you are in these main arteries, then they start decreasing their diameters up to a point that they are now what we call arterioles. In the arterioles, you have muscles. So the arterioles are the structures in our vascular system that's capable of regulating the amount of blood flow that specific areas of the brain receive. And one of these processes that occurs at the arterioles, they occur in order to supply specific areas of the brain with oxygen and with glucose after those processes has happened. The restoration of the PAs, of the action potential, and the IPSP and EPSP. So when you have specific areas of the brain that have an increase, or that modulate their electrical activity, that modulation of electric activity is followed by an increase on the arteriole's diameter close to the place where you had an increased electric activity. And then that blood goes to the capillary system, and then from the capillary goes to the venules and then veins. So all the exchange, it happens on the capillary tree that we have distributed throughout our brain. So how selective is these vasoregulation that happens in our brain? Because one thing that we could think of is that if I have a specific area of the brain that's being modulated, which the electric activity of that area is being modulated, I could think that the whole blood flow of the whole brain could increase. But that's not the case. What we have is something that's selective up to the order of 2 to 3 millimeters, meaning that if you measure the diameter of the arterioles at 3 millimeters from the site where you have an increased activity, that diameter had increased on the order of 5%, as compared to 85% that it increases when you're about 0.1 millimeters from the site of change in activity. So, and the reason why I'm telling you that this is specific, or it has any specificity is that the whole idea behind fMRI is that what you're detecting is that change in blood flow. If blood flow changes all over the brain, then it means that fMRI wouldn't be worth, in a sense that you wouldn't be able to localize an event. What I'm saying is that it has although the spatial resolution is limited, it's still, it's based on an event that has a specific spatial resolution, for say. So the the first two guys that talk about these phenomena, the increased local blood flow as a result of increased electric activity of neurons, were based on observations by Roy and Sherrington and William James. Not the observation of William James, but William James based this phrase here saying that blood flow, the increase in blood flow should follow the increase in neuronal electrical activity. This data here, or this William James phrase comes from data that was, there's this nice paper that was just published about the story of Angelo Mosso. And Angelo Mosso was a very creative physician who decided to test some of these hypotheses. And one of the experiments, he did a first experiment based on invasive measurements. And then he developed this scale here. And the idea was that if you have a stimuli being put to an individual, then you would have an increase in blood flow, the head would weight more, and then you'd get this data in the scale. Well, he got it, yeah. So these are, I'd say, the first measurements showing that indeed we have an increase in blood flow and brain blood flow when you have an increase in electrical activity. I really recommend this paper. It's really nice, really well written, and really nice paper. So just to show you some of the more modern measurements, basically what you do is you stimulate, in this case, you stimulate the palm of a mouse, and then you have an electrode placed in one of where you would expect to have an increased electrical activity, like, and then what you do is to measure the increase in blood vessels around that site. This number here is the distance that this site is from the electrode, and this is how much you'd have an increase on the artery itself. So now what we have is a system that, well, we know that we have a system that would increase and locally increase blood flow when you have an increase in neuronal activity. And now what we need to do is to link that with the MRI. And then, boom, we have the fMRI. So how is it linked? This is linked by, well, part of the story goes back in 1936 when Pauling found out that the magnetic properties of hemoglobin differ depending on the oxygenated state that the hemoglobin is at. Meaning that the oxyhemoglobin is diamagnetic and the deoxyhemoglobin is paramagnetic. What does this mean? Why does it happen? Because once you unbound the oxygen from the hemoglobin, then you expose this iron nuclei that's right in the center of the hemoglobin molecule, and then you confer paramagnetic property to this substance. So what is, for those of you that are, what is it that they're biologists? What's the hobbits, right? So what is it that's paramagnetic and diamagnetic? Basically, is the fact that when you have something that's paramagnetic in the MRI, and the property that's important for us is the fact that when you have something that's paramagnetic, that thing that's paramagnetic, it distorts the field around that molecule more than something that's diamagnetic. That's the only thing I want you to know. So once you have diamagnetic hemoglobin, oxyhemoglobin that's diamagnetic, it distorts less the magnetic field around it as compared to the deoxyhemoglobin that's paramagnetic. And what is it that happens once you have the change in blood flow that follows the neuronal activity? You have an increase in blood flow close to the sites where you have an increase on the electric activity of the neurons. And the blood that's getting there, it's blood that's necessary to supply that place with oxygen and with glucose. So the blood that's getting there, it's a blood that's rich in oxyhemoglobin. So on that site that we have an increase in blood flow, you have a net change on the magnetic properties of that site, because you would have a change in the ratio between oxy and deoxyhemoglobin. So in fact, what happens is that you have an increase in oxyhemoglobin, then you have an increase in diamagnetic material on that region. You have a decrease in magnetic field distortion. And as you have a decrease in magnetic field distortion, you have a light increase in the MRI signal. I'm going to go a little bit farther in this so that we can understand this a little bit better. This was an experiment where they had placed different blood samples with different concentrated of oxyhemoglobin in that sample. And what you see here in blue and in red, I want you to focus on the blue line. This is a MRI physical property that you use when you're constructing your image. So many of the MRI that we have right now are images that are based, their contrast is based on this physical properties. That's T2, relaxation time. But what I want you to look at is the fact that the amount of oxyhemoglobin that blood contains, it's enough to change this magnetic property. So more oxyhemoglobin, meaning that you have this property of the MRI being changed. And this is the property that changed. And this is the property that we detect when we do fMRI. So what is it then? Now what we have is you have an increase in electrical activity, you have an increase in blood flow. This blood flow is rich in oxyhemoglobin. Oxyhemoglobin is diamagnetic. Oxyhemoglobin distorts less the magnetic field. It distorts less the magnetic field. It increases the brightness of the images on or close to the sites where you had an increase in blood flow. This is the story. This is what fMRI is about. This is it's not what fMRI is about, but this is the most used contrast mechanism. This is not the only one, but this is the most used contrast mechanism in fMRI. That's called BOLD, blood oxygenation level dependent contrast. This is the contrast mechanism that we use, and this is the contrast. This is not the only one again, but this is the contrast that we are going to be speaking and using. When was the first time that actually these two things got together? Because this I'm talking about samples, blood samples, put in inside an MRI. So I'm not talking, this is not an image. This is what I'm saying. So when did it become an image? When were the first experiments to show that we could actually use this whole story to localize or to understand or to characterize changes in neuronal activity that happened in the specific parts of the brain? So there are 3 papers that were published in the same year by 3 different groups that showed the same thing. What is it that they showed? Basically, they had a high field MRI. The 3 examples I'm going to show, there are 2 3 Tesla MRI and 1 4 Tesla MRI. In 1902, this was the highest field you had by then, in human MRI. So what was it that they did? They flash a light on a subject's eye for a while, and then they switched it off, and then they flashed it again for a while, and then they switched it off. They flashed it again, they switched it off. So they kept for 30 seconds flashing a light on the individual's eye, and then for 30 seconds the individual was at dark. And then for 30 seconds he would flash again, for 30 seconds at dark, flash again, dark, and so on. So what we are showing here, what I'm showing here is the areas where you had an increase, a net increase in the brightness. The areas where you had an increase in the brightness in response to, as a response of the stimuli. These are the time courses. So what is this? This is the value of the brightness of a specific pixel or of a set of pixels, or an area of the brain. And what you see is while the flashlight is off, you have a specific value of brightness value. And then when you turn this on, it increases. And then you switch it off, it decreases. You turn it on, it increases. The changes that we see here are really, they're very subtle changes. So this is the same thing that happened this is the second paper, and these 2 papers are really well cited, the Ogawa and Kwong paper. But for this Bondettini's paper is not. But it did basically the same thing. But it did with the motor cortex. So instead of flashing a light, he asked the individual to move his right hand for a couple of seconds and then stop moving his right hand for a couple of seconds. So the principle is exactly the same. Unfortunately, this is not what we have. So what we wanted to have is something that when you have a stimuli, you would detect visually, you would detect a change in the contrast of the image. So this would be the dream. You'd have a technique that would have such a high signal-to-noise ratio, the phenomena would have such a high signal-to-noise ratio, that it would be just a matter of conducting an experiment where you would stay for 2 seconds doing something, and then you would do something else. Right away, I would see different regions lighting up, or different regions changing its contrast. However, this bold contrast leads to a very low signal-to-noise ratio. The amplitude of the signal is really low, and the noise, it's low, and it is embedded in noise. So it means that in order to get these maps that are in fact the results of an fMRI study, you have to go through many different processing steps. So it's not a straightforward technique. Once you have the images acquired, you have to go through a whole bunch of steps in order to get these nice maps. I always like to say that this fMRI is a very dangerous technique. Dangerous in the sense that it put together 2 things that are dangerous when they are together. Beauty and ignorance. It is beautiful. But in order to get here, you put your data in a black box. And you can put your data in a black box. And then you'd come out with a p-value that, that would show you something, whatever it is. But it's, the reason I'm telling you that's dangerous, I remember that once I had I was to give a talk in Rio, and I asked a student of mine to go in a scanner and do nothing. So the student didn't, he just went in the scanner, closed his eyes, and stayed there for a couple of minutes. And then I put this image into a black box, like this. So I didn't do anything with the image. I just got the image, didn't pre-process, didn't align, didn't do anything. Put in the black box and got a map. I got something. And then certainly that didn't mean anything relevant to what we were showing. So what we were for the fMRI or for a specific protocol. I'm not going to go through these details. I just wanted to make sure that you understand that this is not straightforward. Every box, like this one, is a black box. So you have different types of alignment, you have different types of pre-processing, you have different type of motion correction, of temporal alignment, of temporal filtering, of spatial normalization, of segmentation, of cortical repair. You have different, all of these are one. Statistical analysis, I'm not going to, this is a big black box. And then you have all these strategies. So there are many groups that this is their research area. So how to develop methods to better interpret fMRI exams. So how do we acquire? The second thing is, the second message here is that since you have a low signal-to-noise ratio, you need something that's robust from acquisition to analysis. So you need to design your experiments in a way that you would increase your signal to noise, or you make it easier for you to analyze the data afterwards. So how is it that the data is analyzed? I'm not going to go into the details. I just want to show you briefly what it means, but by increasing signal to noise ratio or planing your protocol or whatever, because most of the exams or most of the fMRI experiments that are being conducted these days are still based on these type of data acquisition that in the field of fMRI is called fMRI paradigm. So what are the two main types of paradigms, or two main ways that you can acquire the fMRI data? Basically, you can acquire it using what we call a block-designed paradigm. And that is, you have long or relatively long periods of rest that's intercalated with relatively long periods of a task. So you stay at rest for 30 seconds, move your left hand for 30 seconds, rest, left hand again, rest, left hand, and so on. So you, and then task can mean anything. It's a task. It's a language task. It's a memory task. It's an attentional task. It's a whatever task. It's an emotional task. So then you can build up your protocol using this strategy. Using meaning that you're intercalated these long periods of rest with long periods of task. What is the main advantage of this thing? Why people do use this still? Because you have a good detection capability, meaning that this is the best strategy if your question is where things are happening. So if this is your question, this is the type of protocol that you should use. because this increases significantly the signal-to-noise ratio. What is the price you pay? You're paying the price on a temporal resolution. So you're not able to detect different events that happened in a single task. So it's a 30-second task. It's a 30-second that the idea behind it is that the individual is doing the same thing in these 30 seconds. So he's doing the same thing. So in order to circumvent this limitation, there is this second type of protocol that's called event-related. And the event-related task is a protocol paradigm, is when you have the individual for rest for a long period of time, and then he executes a task for a very short period of time, or short period of time. So this is more like when we talk about event-related, the first thing, people that work with humans, the first thing that it's event-related potential. So it's the same strategy that we use in EEG. It's the same thing. The difference is that the EEG, as it has a much greater temporal resolution, these events are much shorter as compared to the fMRI. So here we are talking about a couple of seconds, 1 to 3 seconds of task, and then a period of 20 seconds of rest. What is it good for? So just to have an example, if you want to study like a go, no-go task. What is a go, no-go task? It's a task that's really well designed to detect impulsiveness. So the idea is very simple. You keep pushing a button every time something appears in the screen. As soon as you see something in the screen, you push the button. Unless what shows up is not a geometric figure. So you push, 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 and then all of a sudden a hammer shows up. So what you're to do, you should not push. So you have to control yourself to the impulsiveness of pressing the button. And with this type of protocol, you can look at events like this, like looking at frontal areas that are part of the circuit that's necessary to control this impulsiveness. So this is the type of protocol. Independent of the protocol, how do we analyze the data? What is the type of data that we have? We have the data set is huge because in order to have an fMRI exam, you are detecting long periods of rest with long periods or short periods of task or whatever, but you're detecting that for 5 to 7 minutes. In these 5 to 7 minutes, you are acquiring, at every second or so, you are acquiring the whole volume of your brain. So you're using a special technique called EPI, or ecoplanar imaging. That's a technique that's invented by Peter Mansfield in 1977 to be used in children. So when you do an MRI, you need to avoid movement. And this is very tough to do in a 5-year-old kid, to have them stand still for, or sit still for 5 minutes without moving for a couple of millimeters. So Peter Mansfield, he developed this sequence that is capable of detecting the whole brain, of acquiring images of your whole brain in 1 second. So lately, you can do that, this whole thing here. So you have from top to bottom. You can have that in less than 1 second, 500 milliseconds. And then you acquire it once. And then you acquire it again. You acquire it again. You keep acquiring it for a couple of minutes. So your whole experiment, you are acquiring volumes of data. So we are talking about data that typically are of 64. Each of these here are 64 by 64 with 30 slices. And each of these volumes here is acquired about 100 times. So what is it that you have? You have for each part of your image, for each pixel, you actually have a time series. You have a time series along this direction. And that happens for every pixel of your brain. How is it that you analyze it? So the most simple way to show you how to analyze this type of data, very briefly, is this. So what you do is you have a time series. So you collapse these time series in a single point. So you have an average. And then you have a second average. You do a student t-test, and that's it. Your problem is solved, or partly solved. What is the main problem with this strategy? Is that you're doing that on individual voxels. So you have each voxel here, and you do a t for each part of your brain, for each pixel that composes your brain. And then you look for places where you had a statistical significant difference and so on. But this is very sensitive to motion. If an individual moves for a single pixel, the distance equivalent to a single pixel, so let's say 1.5 millimeter. So the individual moves for 1.5 millimeter means that part of your exam, you're looking at this area of the brain, and the other part, you're looking at the other area. So it's very sensitive to motion. So this is not used anymore. One thing we could think of is, well, let's go then for correlation. So you have a model for your protocol. You knew when the individual was doing the task, whatever the task was. You knew, for instance, here he was at rest for this period of time. And then here in blue, he was moving his right hand. And then rest again, moving his left hand, rest again, and so on. So what you could do is you're expecting an increase in this signal when the individual is performing a specific task. So you have what we call the hemodynamic response function. Basically, it's a model. So we model our task by a specific line or curve like this one. And then you cross-correlate your model with the whole brain, doing a linear cross-correlation, a simple cross-correlation. In regions where you would have something that looks like this, then you would be significant. In regions where you wouldn't have, then you wouldn't be significant. And then you have a statistical map again. This is not being used anymore, but not because of artifact or anything else. It's just because there's something that's a little bit better, that's more general, that's looks like a correlation, but it's called the general linear model. So again, what is the difference between the general linear model and the cross-correlation? Is that the general linear model allows you to have many models inside your model. So you can model the right hand, the left hand, both hands. You can model whatever. And then you model that. You model your time series as a signal plus noise. So this is what you have. This is the problem you have. This is what you have acquired. This, let's say, for a single voxel. For a single voxel, you're saying that the time series of that single voxel equals to your model that you created plus noise. What is it that your model is going to adjust? These values, or the amplitudes of the model. That's it. And then with the amplitudes, you'd have something like this. For instance, here we modeled our data with two independent predictors, or with two independent models. So what we are saying is that our signal is a linear combination of these two models plus noise. If we only have noise, then our models won't be able to adjust the amplitude. So the statistical significance of these models is based on the amplitude of the general linear model, is based on the amplitude of your inputs. If your amplitudes compared to the noise, then it's not a good model for that region, meaning that most of what you have is noise, then this is not statistically significant. And you have a problem that has to do with the p-value. That's something that you guys have discussed already. And the problem is, all right, I have a model, and now I have to put a threshold. What is it? What is the threshold? I'm going to use it. And here, particularly, this is a very important problem because remember that the size of the data set that we are using. We are talking about something that has sometimes 40,000 degrees of freedom. So we are doing many comparisons. So we need to correct for multiple comparisons. And we are talking about something that's around 60,000, the correction. Then what is it, when I choose a p-value, what is it that I change? If I have a p-value like a threshold here, I have a statistical map like this one. If I increase or if I'm more strict, then my map changes. This is one of a typical issue in fMRI. So what is the threshold that you're going to use? Corrected for multiple comparison and so on. Well, so this is fMRI. Then you have those maps. This is what fMRI was. And this is the whole story. This is the base of fMRI. What are the basic applications of fMRI? So the basic application of fMRI, and this is what a lot of people think of fMRI, is a type of modern phrenology. So it's looking at where things are processed or things happen. Or I can't really count the number of times that people have approached me asking for, well, I have this project that I want to know where this happens. So this is in the head, right, yeah. So, and although this is not the only story of fMRI, I have to say that this plays an important role right now. The main role that fMRI plays as a type of phrenology is for a preoperative mapping. So this strategy is becoming more and more available for preoperative techniques. This is just an example of what preoperative mean and why phrenology in these type of problem is important. So this patient here had a ganglioma of about 2.5 centimeters that was close to the motor cortex of this individual, this patient. So once the patient goes to surgery, this is in the operating room. Once you go to surgery, you need to locate some of the landmarks, like the Sylvian fissure, or where is the superior temporal cortex, where is the lesion. So you use an ultrasound, and then you localize the ultrasound, and now you need to, especially for this patient, you need to localize where is the primary motor cortex, or where are the regions where you should not lesion. So basically, what surgeons do is to use this thing here. So it's a pen that delivers an electrical stimuli, or with this strips here that you can slide between the cortex and the dura. So you slide it if you don't have access to the area where you want to stimulate, and then you keep mapping. And once you stimulate the correct area, then you have a response from the patient. Well, the first thing I have to say is that this procedure is not used everywhere. I have to rephrase it. Not many places use this electrical corticogram. So what is it that the surgeon is based on? Experience. So this is something that fMRI have been doing with great success. That is to localize eloquent cortices of these individuals before surgery. Does this substitute the electrical corticography or those invasive measurements? No, it doesn't. But it helps. It helps a lot because on that procedure, on the one I've showed you, I was in the operating room on that day, and the surgeon, it took about 1 hour and 20 minutes to have that place stimulated. And the reason is that you place a pen in a spot, and then you stimulate a pen. It produces a arc, an electric arc. And once you increase the amplitude, it stimulates a little bit deeper, and then a little bit deeper, and then a little bit deeper. And you keep in the same place for a couple of times. And then once you check that there is no response from that spot, then you move, and then do it again. The reason why the surgeon was slipping that strip was not because he wants to show in the movie. It was because he could not find that in that region. So he thought it's not here. So he swept that strip. He stimulated a little bit longer. It was not there. So he came back to the pen, and then all of a sudden he got it. So it does not substitute, but it reduces significantly the time of research. Of surgery, the time of mapping. So it takes much less time. And second, if you want to map eloquent cortex that you need a response from the subject, like language. So how is it done? You do the same thing, but with the patient awake in the operating room. You stimulate Broca's area until the patient stops speaking. So you ask the patient to count from 1 to 20. And then all of a sudden he stops, or she stops. 1, 2, 3, 4. Again. Then you stimulate again. You stimulate 3 to 4 times. You mapped it. So this is even more important for these functions like memory. Then you don't have access. And many individuals like epilepsy patients, they need to go through memory mapping before some of the surgeries that these individuals go to. Another type of, then you can move the ladder a little bit up a little bit, and then try to understand processes that are important to humans, and that you're not able to do it in animal models, like emotion regulation. So are there emotion regulation models? I don't think so. No, right. So this is something that, this is all we have. Is fMRI the something that saves? No, it doesn't. But it allows something. It allows us to study things with this hammer. It's like a hammer. The fMRI is like a hammer. You're trying to break an egg with a hammer, like a huge hammer. So it's brutal. It's brutal, right? It's something. Is it brutal? But although like this type of study, what is it that you're looking for? What is the kind of mechanism that you're looking for? I'm looking for to understand a little bit better the mechanism that's behind a human behavior of controlling themselves, not to respond exclusively with emotion. So I'm angry, I'm not going to fight. I control that. So how is it that fMRI have been contributing to this? So in humans, what we see today, for instance, is that if I look at the signal in the amygdala or in the frontal cortex, I'm capable of trying to dissecting this process. And what is it that this guy is showing here is when you reappraise something. So this is a stimuli that's being used a lot in emotional regulation protocols. And that is, I show you an image of a mutilated body. And then you have, you feel the, you have the feelings that that image brings. And then I'll tell you, don't worry, what I'm showing you is an image of a movie that played a couple of days ago. So this is not science fiction. This is not real. So this is a cognitive strategy that we can mimic in the lab, and that we used it. A lot of times use the cognitive strategy of reappraising, of trying to of doing this reappraisal, of reevaluating that situation. And then that's for good. I mean, I should reevaluate that. And then cognitively you do that. What is it that people have been finding is that when you reappraise, indeed you have a decreased signal from your amygdala and an increased signal from the prefrontal cortex. So this is the type of study that you could do. Another example that, and this is an example that we have been working in lately, is try to understand something that's very human, that is capable of reporting, of telling specific states of consciousness. So one thing that we are interested in the last couple of years is to use these techniques to try to understand a little bit better the neural basis of the experience induced by hallucinogens, by psychedelics, like the one that we have been using, studying, that's the ayahuasca. I'm just going to tell you, I didn't bring, just brought the slides of what we've done. So what is ayahuasca? Ayahuasca is a psychedelic beverage that's being used for many years traditionally by Amazonian Indians. And this substance, ayahuasca, is used in many rituals. It's used in rituals that individuals go to war. It's used in rituals where individuals want to have a revelation. It's used in ceremonies where individuals want to get cured of something, so medicinal purposes. And in some Indians, ayahuasca is the main pillar of that culture. So it really plays a significant role in that culture. It is the spinal cord of that culture. In the beginning of the 20th century, this beverage was brought to urban centers through the Amazon region, and then now it's spread all over Brazil, mainly in two different sects, the Santo Daime and the União do Vegetal. So these are two religions that use ayahuasca in their ceremonies. In Brazil, it's legal to use it for research purposes and as a religious, in religious ceremonies. So there are many effects. I mean, when we look at the effects of ayahuasca using the neuroscience view of neuroscience, what you have is you have a very strong modulation on the visual system. So it's an it induces hallucinations, visual hallucination mostly, but auditory and different senses. There are different senses that are modulated by ayahuasca. It also increases the perception, the interoceptive attention. So it's not a cognitive experience in the sense that you're not capable of controlling it, or saying, well, I'm gonna do this or do that. It's a totally altered state of consciousness. So with this substance, we decided to try to understand the neural basis of some of these processes. The first process that we looked at was the visual mental imagery system. So basically what we did was to have a couple of individuals, 10 experienced individuals from one of these churches, to go through a very simple protocol. They would have an fMRI protocol similar to the ones that you've guys seen. In some periods, they would have a visual perception task where they would see complex images like this one. They would go through a mental imagery task, and during the mental imagery task, they had their eyes closed and they had to imagine this image here, and if something got brighter in their mind, so if they saw something that's different from the image, the seed image, they should follow it and then and see it. And then they would go through periods where we would have the scrambled version of this visual perception image to serve as a baseline. What is it that we found? So this map here is a map that's a little bit, so this is a p to the 9 minus 9. Yeah. Right, yeah. That's whatever, right? So, what is it that we found? Basically, what we found is that this is what we call the conjunction analysis, meaning that we are trying to exclude effects that are not related to a conjunction between the T intake and the visual mental imagery task. So what is it that we are excluding? Whatever I'm showing here, so this area lighted up. So this area is an area that supposedly is not involved in the perception task, and it's not involved in, it was not modulated by the, without the T. So what we're showing here are the structures that were modulated by the T in conjunction with the task. So they were modulated by the task. The individual had to be engaged in this task. And what are these structures that we found? Basically, visual areas, including the primary visual area. And this is for the neuroscience community. This is something that we have been doing a follow-up, because there is this nice discussion in the literature if a visual hallucination come with the modulation of the primary visual cortex. So there are some papers saying that it doesn't. There are some papers saying that it do, that it doesn't, that it does. So what we are showing here is that in this case, it seems it does. So the primary visual cortex and many different structures of the visual cortex get modulated by the T and the mental imagery. Mesiotemporal areas like the parahippocampal gyrus, part of the hippocampus, the fusiform gyrus. So these structures are also modulated, and these frontal polar area that is also modulated. So what we see here is that it seems as though that ayahuasca is capable of or doing this mental imagery task of modulating parts of your visual system and parts of your memory system. So one speculation that we are after right now is to came up from this study, and that is that maybe what ayahuasca is doing is bring light to your own experiences. So give your own memories a sense of reality through your visual system. So this is just a speculation, and this is something that we are looking right now. But just to show you how this phenomenon is robust. So these are the time series of some of those structures. I just want you to focus on BA17. That's the primary visual cortex. In blue is the signal, so it's the average of the signal of the primary visual cortex when the individual is with their eyes closed, imagining something with their eyes closed without the ayahuasca. So why is it that it's negative? It's negative because the baseline is the scrambled version. So the baseline, he had his eyes opened, looking at an image, a pattern, a visual image. So you would expect that it modulates the visual cortex. And in fact, it does. So if you look at the imagery part, when the individual is with their eyes closed without the ayahuasca, then you expect a negative signal as compared to the scrambled. All right? If you look at the other three, what do they represent? Perception. So the perception of the task when the individual is with their eyes closed, with their eyes open, with and without ayahuasca. So the ayahuasca doesn't modulate the primary visual cortex in the perception. So meaning that the primary visual cortex doesn't respond differently to what it perceives with or without the ayahuasca. But if you look at the red line, and that is the mental imagery with ayahuasca, what we are showing here is that in all these structures, the signal of these structures, when the individual is with their eyes closed, imagining something, is similar to when he's with his eyes open, looking at something. So the second speculation is that they're seeing. So this is the type of study that you can do with fMRI. But all right, most of what we saw, and most of those protocols that I've shown you, they intercalate periods of rest with periods of tasks. So most of the fMRI protocols, most of them, what they do is they ask you to do a task and then they ask you to stay at rest. What is the assumption? Is that when you're at rest, your brain is doing nothing, right? That's the assumption. I mean, you're saying, but what is it that it's doing at rest? And then I'd say that this have changed the way that we look at fMRI, in the sense that one thing you can do, and one thing that most of the groups do, is to keep the system as free as possible and not interfere with the system. Because when we do that task rest thing, what we're doing is inspecting how the system responds to something, right? So it's a response function. That's what we are investigating. And by looking at this, then you can kind of look at the system when you are not interfering with it. So what is it that the brain is doing at rest? And that gave rise to what is been called resting state fMRI. What is resting state fMRI? Basically, is a very simple protocol that you have an individual inside an MRI scanner doing nothing that I ask for a couple of minutes. So he's just there and I'm acquiring data from his brain. And why is it that this strategy has gained a lot of space in the fMRI community? is because a lot of things started to have, it brought a meaning. It had a meaning. It was consistent. So the type of finding that you have with this type of analysis, it starts to build out confidence in the fMRI community. And what is it that you have? You still have time series like this ones. So you have time series like this blue that comes from this area. You have yellow. You have whatever. So you still have time series. But you have time series without the interference. You have a resting state time series. All right. So what was the first thing that people did with it? They looked at if this strategy was capable of understanding somehow how is the brain functionally connected and not structurally connected. So if these time series could bring the information that this specific area is linked functionally to another area of the brain. So as a proof of concept, what they did was, and it was funny, this was published in 2000, and it took about 8 to 9 to 10 years in order for this to really spread in the community. So what is it that these guys did? They had a resting state in an individual. So you had an individual for a couple of minutes inside the scanner doing nothing. And the same individual would do a motor task. So he would do a finger tapping with the right and left hand, for instance. And then he would do all that analysis that I've shown you, and this is the result. So the motor task would elicit these areas, these expected areas, primary motor cortex, somatosensory cortex, cerebellum, and so on. So they got the same individual. They put the individual and did a resting state. This time, this guy's not doing anything. He's not doing a motor task. And now, they place a seed in the primary motor cortex. So they localize the primary motor cortex of the individual right here. They have the coordinates. Now they grab this coordinate and they bring the coordinate here in another exam. And what they did here was they got the time series and they correlate this time series with the rest of the brain. What is it that the motor system? So a great part of the motor system, you can, and this is the reason why people have been calling this functional connectivity. So it's a way of trying to generalize how different areas of the brain are functionally connected. So it's how they do co-vary. If one area, so you're doing a correlation between whatever area and whatever area. And then you have, what is it called, the resting state networks. What are the resting state networks? Resting state networks are a number of networks. You have the salience network, you have the default mode network, you have the auditory network, you have the basal ganglia network, you have many different networks that showed up using this strategy. What is it? You place a seed in a specific area, and then consistently for many different individuals and for many different areas and in different countries and whatever, you have a specific network that whenever you place a seed there, it will be functionally or correlated to these other areas. One of these structures is what's called the default mode network. I'm not going to go into the details of the default mode network, but basically this is one of the resting state networks, and apparently the reason why it is here is because of my question. Apparently this is a network that's very important when you're at rest. So when you are at rest, apparently this network, the default mode network, gets engaged. So a lot of people have been trying to understand the meaning of the default mode network. What is it? What is it that you're doing when you are at rest? Mind wandering. Or not at rest. I mean, a lot of you are mind wandering right now. So it is, and then this structure changes with meditation. This structure changes with ayahuasca. We've just shown that it changed with ayahuasca. It changed in different diseases, like Alzheimer, schizophrenia. So this has also changed in different sleep stages. This really opened this new perspective. And it opened a perspective of looking at the brain it's one way to look at the brain as a complex system. One thing that a lot of people are doing is to do the following. So I will segment my brain in 106 structures, like this. Each of these structures, so it's segmented based on atlases, on anatomical atlases, on functional atlases. You choose. This example here is for the 106. It's split the brain in 106 areas, 53 in each hemisphere. Each of these areas here, I'm going to call it a node. And then for each node, I'm going to average the time series of each node, so of each pixel for this node, and then I have a one-time series per node. And then, yes, my 106 ROIs are specific areas anatomically defined. And for these areas, I have a couple of voxels inside, and then I average these voxels inside this area, and I have one time series. So for each one of these 106 areas, I have one time series. And what I do, I correlate them in pairwise. And then what is it that I do? I create the links. So then I have the time series, and the correlation values are the ones that I'm gonna try to link to different nodes. And then I can do or extract the network topography of these networks. So this is something that we are doing with ayahuasca, for instance. We are doing with schizophrenia. We are doing with treatment of schizophrenia. So one problem of these, of people that work with this, Mauro, Natalia, Gigi, so just is to find a meaning. All right, I have a change in the clustering. So what does it mean? And this is exactly where we are. So does ayahuasca change some of the characteristics of this topography? Yes, it does. So for instance, it changes the path length. So you have an increase in path length, and you have a decrease without a change in clustering coefficient. So who is looking for a meaning here? Ganji. Ganji Vishnavata. So what is Ganji idea? That's, to me, it's a nice idea. If this result here may represent the following. You have, say for instance, just to mention two clusters. You have two clusters. Three clusters, just to make things easier. You have three clusters. These three clusters have one link between them. So these clusters link to these clusters by one, these clusters to this one by one. So the path length is short. as compared to this. Because now, I have to go through this way without changing the clustering coefficients too much. So, it makes sense, but I don't think we're quite there yet. No, there are different ways. So, anyways. So, just to wrap up, what is, I'd say one of the frontiers that fMRI is at right now is using these strategies, using these resting states or long time series. And most of the times what you have is you don't ask the subject to do anything. Most of the times what you do is to train the subject for long periods of time with a specific stimuli or sometimes a task. And then you look at the stimulus and you train your time series to a specific stimuli. So you have a specific stimuli, and that specific stimuli is represented by a time series. And then you train your network in order to understand what are the features of your stimuli that are detectable by your time series. So once you have, for specific categories or for specific images or for specific auditory stimuli or whatever, your goal is to, from your time series, your goal is to try to estimate or to decode what was it that the individual was looking at, or what was the part of the task that the individual was in. Just to make it a little bit more concrete, this is an example, and this is an example that we have been working with right now. We present, we have 6 categories: cars, trees, reptiles, food, people, and houses. So these are the 6 categories that we have. So we show the individual one image of one category for 6 seconds. And then he has rest, and then another category, rest, category, rest, category, rest, category. This is shown for 6 seconds and rest for 6 seconds. We did 24 runs of 60 images. So that means 12 hours of scanning time. So 24 runs of half an hour, and each half an hour, the individual is looking at this. Mixed up, different images. So we have 60 images, 10 of each category, and we keep showing the individual for a couple of hours, 12 hours. All right. Now, what we do is we train this network for each individual category. And what is the next step? And this is where we are right now. The next step is to leave one out. So what we do is we train, we leave one category, one image out of this category, and then we present one of the categories, and the algorithm had to guess what was the category that I've just played. So I train with leaving one run out, One of the 24 runs I leave out. I train with 23. And then I get this 24th. I put in my machine, and then it guesses what category is that one. Clear? This is for one. What is nice about this strategy? I have to say before I show you this first results here, it's that most of the neuroscience that's done in humans, they are based on statistic inference of groups of individuals. So what I'm saying most of the times from a neuroscience experiment is that I have 3 rats, I have 5 mouse, I have 2 monkeys, I have 10 individuals with depressive symptoms, and then I compare groups, right? This is our strategy. Our everyday life is this. What is nice about this is that I'm looking at a single individual. So this result here doesn't come from groups of individuals. It comes from a single individual. I'm putting the individual. So this type of paper, with fMRI, you need something like a magic number of 20 individuals. With this, 3 is enough, because I'm talking about 12 hours per each individual. And this opens a very interesting perspective in neuroscience of humans. That is look for the variance and not for the average. This might be a strategy that we might use to look for the individual, and that is to acquire a lot of data from the same individual instead of acquiring less data from a great number of individuals. That's just another strategy. And this type of design is really interesting in part because of this. So what does this graph mean? Mean that when I left one out, these are the results for 2 subjects. So we did already 2 subjects. When we leave cars out, for instance, he get right on 11 of the 20 runs. So when I contrast that to each run, when I do for every run, it gets right in 11, 15. What's interesting is that for humans, in both subjects, they detected 20. They got 100% right. And just so that you know that although 11 and 20 might seem a small number, the chance in this case is 3.3 over 20. So it's not bad. But there is a trick. So we are doing our first steps here. And what is the trick here? Is that I'm training my network using the GLM method. So I'm helping already my method. That's why I got these things here. So just to finish, I hope I have convinced you of why fMRI have lived for 20 years or a little bit more than 20 years. If it was just because of the phrenology, if it was a technique that would be used just to map the brain as in the phrenology philosophy, it probably would not have survived this long. But there are still a lot of issues and a lot of things that I believe that fMRI will still be there for the next couple of years. If you look, for instance, at perspectives of doing direct current measurements with MRI, this is a perspective. I'd say that this is a perspective that has been a perspective for 20 years. But still, it's there. So it means that instead of relying on the bolt signal, like we're doing, instead of relying on this epiphenomenon of the neuronal activity, we would use the MRI to look at the electric activity itself. So MRI would gain significantly if we have this going on. Increased signal-to-noise ratio. This is something that it's already, I'd say that's already at the maximum rate. There is this 11.7 Tesla scanner, human scanner, being built in France at the Neurospin. And what we know already from, and also from the physical point of view, is that from the physics, is that although you have an increase in signal-to-noise ratio as far as you increase the magnetic field strength, you, and this is pretty much linear until you reach 3 Tesla. After 3 Tesla, the thermal noise increases more rapidly than the gain you get with the increase of magnetic field. So that means that the 11.7 Tesla is not 10 times better than the 1.1 Tesla. It is better, but it has a lot of problems, a lot of susceptibility issues, a lot of problems. So I'd say that 3 Tesla, 7 Tesla, the most, would be something that we will see in the next couple of years. And then, again, all the resting state stuff open this new perspective of doing effective connectivity, for instance, because the functional connectivity that I've talked about, of doing the correlation between 2 brain sites using this resting state strategy, you cannot infer causality between these 2 sites. This is something that's a lot of people have been working in lately to come up with better methods to infer on effective connectivity, and that is to include the causality on these models, on these nodes, and trying to understand what the resting state or the functional connectivity means. What is it that connectivity between 2 structures like we're detecting with fMRI, what does it mean? And last, the decoding stuff that's been gaining a lot of space in the last few years. So I'd like to thank you Siddhartha, thank you Gigi, thanks the Institute, International Institute of Physics. Thank you for all the organizers, and I hope you have enjoyed it.
At what point do the gradients that you need, you have to send pulses of all stuff. At what point does it start to be dangerous to people? Because if you put 11.7 Tesla, it's not a problem, but you have to put all the other gear that changes magnetic fields and creates currents.
Right. Well, depending on how you encode phase, the 1.5 Tesla already is capable of inducing currents in peripheral nerves. So it's a technology that's moving in parallel to the increase of the magnetic field in order not to induce a peripheral nerve or induce brain stimulation. So what I'm saying is that in a 1.5 Tesla, depending on the strategy that you use, you are already going to induce peripheral nerve. So there are different strategies on because what is the gradient? Basically, what it's going to do is to
resolution.
The resolution, right? The spatial resolution of your image, and with the higher field, then you don't need. You have a lot of signal already, so you can reduce time for your or you have less time for your acquisition, because it's always a trade-off of space and time, right? So if you have the signal, from the 1.7 Tesla, you can use that for space or for time, and then depending on how clever you are, then you're not going to use that in the sense that in a way to stimulate the peripheral nerve. So it's not only a matter of the intensity of the field or the intensity of the gradients, but it's a matter of how this technology is built inside the scanner. So if you have that in 1.5, and then you're able to circumvent that. But on the other hand, I don't know if there is a magic number for the gradients, that you need a gradient that's less than this and apply it on the AP direction, because otherwise you would induce, inevitably, you would induce currents.
I want to ask about how we can make a connection between EEG source localization and fMRI network connectivity in resting state.
Right. There's only, that I know of, there's only one strategy. Well, first you need to acquire both simultaneously, right? Do the EEG fMRI measurements. And then what some people do is to use either information as a restriction for applying the second method. In the other, meaning, so I can use the fMRI information to limit my inverse method to localize with the EEG. So I don't know of a method that use independently both signals in order to gain spatial resolution independently. So what I know is one constraining the other. So you have the constraint of the fMRI on top of the EEG, or the other way around.
As I know, the resting state fMRI returns to gamma band of the brain. It's something to somehow.
Right, yeah, that's...
Is it possible to make connection between gamma band in EEG and also in resting state? But ineffective if we want to find more distance between two regions of interest, maybe we should focus on alpha or other waves to synchronization between two different areas of the brain.
Right.
So
you mean on the EEG,
yeah,
on the EEG side?
So I want to know, is there any study in this regard who make connection between these kind of effective connectivity and also localize the gamma bands and also alpha relation to fMRI, resting state fMRI?
Not that I know of. Maybe Gigi. Oh, Gigi's not here. Because he might have known, but I don't know.
It's a hyperpolarized molecule. I heard about lung magnetic resonance.
Right.
But this is brain magnetic resonance.
People
so
are trying
different.
to do the same thing, tagging these hyperpolarized molecules, two molecules that has a biological significance, to do something that's similar to what PET is doing. So you try to tag a glucose molecule with a hyperpolarized molecule tag to it, and then you would be able to detect that on the MRI. So it's a strategy that the MRI would give some of the information that the PET inspect gives us today.
Your classifier mix the reptiles with food, right?
Right, one of the subjects. Yes, yes. There's a discussion in the lab. Who's this guy?
No, I mean, just a naive guess. It just looked like the two have some kind of effective valence to it. I mean, you dislike reptiles usually, and you like food usually.
Right.
And the other ones are kind of neutral. So have you just looked at signals or the hotspot or something? You know?
No, we haven't. But the reason why we've chosen these categories are these are the categories that apparently are the ones that are most far apart in space. So this is the reason why we've chosen this one. The reptile, the reason is because of the ayahuasca. Because ayahuasca visions, a lot of them are of reptiles for some reason. So this is the reason why it is there.
Do you know any work that use decoding methods with EEG signal?
There is, but then they are not as effective as the fMRI because of independency. So the fMRI you have a much better independency on the results than on the EEG.
So I have a trick question for you, but I think it's also a good question for the students. So if you had an unlimited supply of resources,
right,
what would you do?
I would go to space.
I thought that was easy with ayahuasca.
Well, do I need to tell everyone? I'll tell you later.